So hello students, welcome again. So today we are going to discuss about the single cell protein part second. So in this last part we have discussed about the single cell protein from the production of SCP from the algae, that is spirulina, and its production methods here it will be discussed. And today we will discuss about the production of SCP from the fungal biomass, that is yeast. So the species of yeast is cultivated to produce protein as Saccharomyces, and Candida is there, that is the food is, then Tropina is there, that alkane yeast, which has about 11.2% of total nitrogen, 72% crude protein, then 6% lysine and the 4% of threonine per we can say 100 gram of protein. Okay, so digestibility of crude protein is about 91%. So the former thrives on we can say sugary solution of molasses, while the later grows on the starch. So yeast are capable of assimilating ammonium salt. So sometimes nitrates also we can say as a source of nitrogen into protein, and assimilation of a variety of organic substance as a source of carbon. So there are methods of production of this yeast. So the molasses is placed in the fermenter diluted with the water and then pasteurized. So then we can say compound added to this solution are ammonium sulfate is there, then we can say diammonia is there, then hydrogen phosphate, magnesium sulfate, biotin, thiamines. So or we can say yeast extract is in traces to that. So the fermenter is then inoculated with the small amount and desired strains of the yeast. So the sterile air is driven into the fermenter by the motor driven we can say propeller pumps is there. So the cells of the inoculum utilize the nutrients in the medium. So they grow very fast, consuming the nutrients in the substrate. So because of the growth of the yeast is very fast, so doubling in every hour we can say. So large amount of substance is needed to be added to the fermenter from time to time. So the aeration rate is adjusted according to the pace of growth. So the chemical reaction of the growth and the aeration produce a foam on the surface. So a substitute we can say anti-foam agents like polypropyl glycol is being used, are being added. So heat of this chemical reaction is also generated. So therefore it is essential to maintain the temperature of the ferment at that is about 30 degrees Celsius. So optimum pH should be maintained, that is 4 to 4 we can say 4 to 4.5. Ammonium sulfate is used as a buffer to maintain the pH. So the fermenter is operated in we can say in the two techniques, that is a batch technique and the second is that is a continuous culture technique. So in the batch technique, in this technique the fermenter is evacuated at the end of the process. So the fresh culture and the substrates are then added and the process is repeated as required. Whereas the continuous culture process we can say here the process goes on without stoppage. Okay, so the technique is more demanding. So both of these techniques have their advantages and some disadvantages. So yield of this yeast is about 50 gram per hour per liter of the substrate. So this is the process of production. Now what are the factors affecting the production of the biomass? So organic substrates and the nitrogen we can say ratio, that is C ratio, and favoring high protein content should be between 7:1 or 7:1 or the 10:1 and the 10:1. So the pH of the nutrient should be about 3.5 to 4.5 in the minimum growth of the bacterial contaminants. So the temperature of the fermenter should be, that is a 34 degrees Celsius or the same strains it is 40 to 45 degrees Celsius. So oxygen for the growth on carbohydrates should be 1 gram per gram of the 1 gram per gram of the dried cells, and the growth on alkanes should be about 2 gram per gram dried cells. Okay, then strict maintenance of this whole process, continuous maintenance should be done throughout the process. Okay, so these are the factors affecting the yeast biomass. Now, what are the recovery of the yeast biomass as protein? We can say so. Yeast cells are small in size. We can say about 5 to 8 microns, and the density is very. We can say density of each reaches to 1.1 gram per ml. So the yeast cells are recovered by decantations, centrifugation, including washing and drying treatment methods. So after washing, undesirable trace of medium are removed, which are again recycled for the economic reasons we can say. So after drying and final harvesting by rotary vacuum filter, yeast cake containing 20 to 40% dry matter is obtained. So it is then we can say properly dried to get yeast product of we can say 60 to 10% of the content. Okay. Now, what are the importance of single cell protein? So, as we all know, the protein single cell protein producing capability about 250 kg of cow and 250 gram of microorganisms are often compared to understand the economic situation of this single cell protein. Okay, so basic will go, that is a single cell protein is a protein rich and can be stored and shipped over long distance. Okay, so they are best sources of animals and animal fodder as well as for the human food also, and replace other proteins rich material such as soybeans meals or the fish meals. So the microbes can be easily handled and subjected to genetic manipulations so that novel proteins products can be obtained. Okay, so we can say they are truly biological in origin, SCP, even through produced industrially. So these processes don't imbalance and natural ecosystems. But so that by single cell protein production, no novel synthetic compounds are produced, and the technology being based on recycling of waste and pollution free, bio-friendly, and it is an eco-friendly one. Okay, now the next, that is the raw material necessary for single cell production are available as a natural biomasses or industrial or agricultural waste that are available in plenty and cheap for. So the wide range of raw materials that are otherwise wasted can be fruitfully used for the single cell productions. So this next, that is a microbes grow at a very fast rate in optimum culture conditions. So some microbes double their biomass as lesser than 30 minutes. So we can say the culture conditions and the fermentation process are simple and economical. So to supply the needs of increased the human population and scarcity of protein, the only we can say solution is to use microbial protein or SCP as a cheap cost one. So these are some importance of the single cell protein. Now is the acceptability of the single cell protein. Well, it is very difficult for the common person to make a habit of using this protein. So the first, there are following limitations for the we can say single cell protein, that the nucleic acid contains. So it is very high in microbes. Example, we can say 4 to 6% in algae, 10 to 15% bacteria, then 5 to 10% in yeast. So this is highly dangerous as a human have a limited capacity to we can say degrade nucleic acid. Okay, then carcinogenic and other toxic substance. So in this single cell protein have certain carcinogenic and toxic substances. So they include hydrocarbons, heavy metals, then mycotoxins and some contaminants. So the nature and production of these compounds depends on the raw material and the types of organisms used in it. Next, that is a pathogenicity. So there is a possibility of contamination of pathogenic organisms with the SCP. So digestibility, so the digestion of microbial cells is rather slow. So this is frequently associated with indigestions and allergic reactions in individuals. So the cost of the cost with things. So the food grade single cell protein production is rather expensive than plant sources of protein. Example, soya meal. So the single cell protein for human consumption is 10 times more expensive than the single cell protein for the animal feed. Okay, so the choice of the substrate for single cell production has more importance for its acceptability. So the substrate used for single cell production for animal is waste of sugar or the dairy industry. So the choice of organism will partially determines the process, technology and the nature of the product. Okay, so thank you very much.